Okay, so let's unpack this. Imagine taking like a tiny scrape of skin, right? Reverting those cells back into this completely raw, unspecialized state, dropping them into a petri dish, and then
just, watching them physically build themselves into a fully functional, life-giving organ.
right, without giving them a blueprint. Like, you don't use a single drop of embryonic tissue. You literally just provide the raw biology, and these cells somehow figure out how to organize themselves.
They form this highly regulated assembly line. I mean, it sounds like pure biological science fiction. We're basically talking about asking a microscopic pile of bricks to, like, spontaneously assemble into a skyscraper.
Yeah, complete with working plumbing and specific rooms and a workforce.
Exactly, just from being placed in the same zip code, essentially.
And yet, that is the reality we are looking at today. So welcome to this deep dive. Our mission today is to explore this truly mind-bending 2026 paper. It was published in the journal Science by Yoshino and this incredible team of researchers.
The landmark paper really changes everything.
It does. We're going to examine how these scientists successfully built a fully functional testicular niche from scratch, using mouse pluripotent stem cells to create life-giving sperm completely in vitro. So if you've ever been curious about the cutting edge of reproductive biology or, just wondering how far stem cell tech has actually come, this deep dive is going to fundamentally change how you view the dawn of biological life.
Absolutely. But to appreciate the sheer scale of engineering this factory in a dish, we really have to look at how nature built it in the womb first.
Right, baseline.
Yeah, we tend to view biological sex as this rigid, unchangeable trajectory right from the moment of conception.
Yeah.
And genetically, I mean, that's true. Whether an embryo is XX or XY is locked in the millisecond fertilization happens.
Okay, but the physical embryo itself tells a very different story early on, right? Like, it's sexually indifferent.
Completely. It is a true blank slate. If you look at an early embryo under a microscope, there are no physical clues indicating what it's going to develop into.
None at all.
None. The architecture is totally neutral. And the main characters in our story today are what biologists call primordial germ cells, or PDCs.
Okay, PGCs. Got it.
These are the precious precursor cells that will eventually become either sperm or eggs. Now, in mice, these cells pop into existence around embryonic day 6.5, and this is key, they don't start in the gonads.
Oh, really? Where are they?
They're essentially just floating around. They have to physically migrate through the developing embryo to reach their final destination.
Okay, so I'm picturing these germ cells as like passengers sitting on a train approaching a massive switch track.
I like that analogy, yeah.
The passengers have absolutely no control over where the train goes, right? The surrounding environment, the tracks themselves, are going to decide whether that train gets routed to Testistown or Ovaryville.
That's spot on. The germ cells are totally at the mercy of the surrounding tissue, which we call the somatic cells. Those somatic tracks don't start shifting to lay down a specific route until around embryonic day 12.5.
Okay, 12.5.
Right. That's when dimorphic or two-path differentiation kicks in. And the mechanism driving this split relies on a master switch gene located on the Y chromosome. It's called stray.
And so if the embryo is XY, that stray switch just flips on.
Exactly. And when stray turns on, it triggers a cascade. Its main target is another gene called SOX9. You can think of SOX9 as the foreman of a construction site.
The foreman.
Yeah, it aggressively directs those neutral surrounding somatic cells to become Sertoli cells.
Right.
Sertoli cells are the foundational pillars of the male reproductive environment.
Okay, but what happens if you have an XX embryo? Like, there's no Y chromosome, which means no stray gene to flip that master switch.
Right, so without stray and SOX9, a completely different set of tracks gets laid down.
Yeah.
Female pathways driven by genes like 1,4 and ARSPO1 take over the construction cell.
1,4 and ARSPO1, okay.
Yes, and they utilize a signaling route called the beta-catenin pathway to drive ovary development. But what we're looking at here isn't just passive building. It's a mechanism known as mutual antagonism.
Mutual antagonism. That sounds like two roommates who actively sabotage each other.
That's basically what it is. It's a biological tug of war.
Mm-hmm.
These genetic pathways don't just build their respective organs in isolation. They actively fight to suppress the opposing pathway.
Wow.
Yeah. The male genes produce proteins that bind to and silence the female genes, and the female genes do the exact same thing to the male genes.
So it's like an evolutionary safeguard.
Exactly. It prevents intersex characteristics, ensuring the tissue commits 100% to one functional path to guarantee successful reproduction.
So it's not just build a testis, it's build a testis and actively burn any blueprints you find for an ovary.
Huh, exactly. Burn the blueprints.
Okay, let's pause for a quick recap before we move into the actual laboratory stuff.
Yeah.
If you're keeping a mental checklist, early embryos are physically neutral. The germ cells are just passive passengers, and the environment decides their fate through this brutal, mutually antagonistic genetic tug-of-war triggered by the SRY gene.
Perfect summary.
All right, so with that foundation, let's look at the actual experiment. For years, scientists have known how to make the passengers the germ cells in a lab, right? We can coax stem cells into becoming primordial germ cells pretty reliably.
Yeah, we've had that part down for a bit. The real roadblock holding up the entire field of reproductive biology was figuring out how to build the tracks. We had the passengers waiting on the platform, but nowhere to send them.
Right.
Recreating that complex surrounding somatic environment, the testicular niche entirely in a petri dish, that was the holy grail. So the researchers approached this by taking mouse embryonic stem cells, but they needed a way to watch the microscopic tracks being built in real time.
How do you even do that?
They utilized fluorescent reporters.
Wait, are they altering the DNA of the stem cells to make them glow? I just want to make sure I understand what a fluorescent reporter actually is.
Well, they aren't changing the fundamental function of the cell's DNA. Think of it like wiring a security camera to the factory's genetic power grid. The researchers spliced a fluorescent tag next to the SOX9 gene, remember? That's the male formin gene.
SOX9.
Whenever the cell turned on the power to SOX9, the camera would light up, making the cell glow green under a microscope. And they did the exact same thing for the FOXL2 gene, which is a major marker for ovarian tissue. If FOXL2 turned on, the cell would glow red.
Okay, so we literally have biological traffic lights here. Green means the testes blueprints are running. Red means the ovary blueprints are running.
Precisely. And the goal was to drive XY stem cells through that indifferent bipotential state we just talked about and push them toward becoming what the researchers named testicular somatic cell-like cells, or TesLCs.
TesLCs, got it. And how did they push them?
To guide the cells, the researchers modulated specific chemical signaling pathways, specifically the BMP and WNT pathways at very, very precise times.
Okay, I have to push back here for a second because this sounds a little too much like magic. They just poured some BMP and WNT chemicals onto a clump of stem cells and they magically organized into complex tubules.
It does sound like magic, I know.
How do we know these aren't just green glowing imposters? Because just turning on one single gene doesn't make you a fully functioning biological environment.
And the researchers anticipated that exact skepticism, right?
Yeah.
Relying on a green light isn't enough proof for a breakthrough of this magnitude. So to verify the architecture, they utilized single-cell transcriptome analysis.
Okay, so if someone is hearing the word transcriptome for the first time, how should they visualize what that is actually doing?
Imagine walking into an office building and being able to instantly read every single email, memo, and sticky note being written by every single employee at that exact second.
Wow, that's comprehensive.
Yeah, the transcriptome is the complete readout of all the genes being actively expressed or read in a cell at any given moment. By capturing this data, the scientists proved that the lab-grown cells weren't just randomly flashing green.
So what were they actually doing?
The cells were enriching a whole suite of vital male genes. SCARI, SOX9, AML, and DMT1. Their development closely mirrored the exact in vivo real-life sex determination process.
So it's like checking the cell's browser history to see what it's been studying, and the history is just completely full of testis construction manuals.
Exactly. And it translated into physical architecture, too.
Yeah.
The cells physically organized into seminiferous tubule-like structures.
Like actual 3D structures.
Yes, they formed the exact spatial arrangement needed, complete with Sertoli cells ready to nourish sperm and interstitial-like cells surrounding the tubules.
That is wild. Time for another quick recap, I think.
Yeah.
So the scientists used glowing tags to monitor the cells, bathed them in specific chemical signals to guide their development, and used transcriptome analysis to prove they had genuinely built the male reproductive architecture.
Right.
So the factory is built, the green lights are on. Now they just need to introduce the workers, the germ cells. But from what I understand, when they tried to get the workers inside, the entire system essentially just collapsed.
It did. This is where they hit a massive biological roadblock. So they have this beautiful, intact testicular organoid, and they have a separate batch of lab-grown primordial germ cells. To mix two distinct cell types in a lab, the standard procedure is dissociation. You take a TesLC organoid, use enzymes to gently break the cellular bonds apart, mix the germ cells into the slurry, and just let them all clump back together.
So you tear the building down slightly to let the new people inside and then rebuild it? I mean, that sounds totally logical.
It does, but when they broke the cells apart, the cells suddenly started glowing red.
The red lights turned on, the ovary alert.
Exactly. The FOXL2 gene, that definitive female marker, was suddenly de-repressed. Just by separating the cells from one another, they began reverting to a female ovarian state. And remember, these were strictly XY cells.
This highlights a staggering biological asymmetry mentioned in the paper. Like, to get those green male SOX9 cells, you strictly need a Y chromosome.
Right.
You cannot force an XX cell to become a male cell in this specific developmental window. But you can absolutely generate female red FOXL2 cells out of XY cells.
That's wild.
Here's where it gets really interesting. The male developmental state is incredibly fragile. It is constantly fighting to stay male, whereas the female state is the default baseline that the cells want to snap back to if they're given the chance.
Yeah, and if we connect this to the bigger picture, to figure out why breaking the cells apart feminized them, the researchers went back to the transcriptome analysis. They looked at the emails and memos of the cells after dissociation.
And what did they find?
They discovered that separating the cells severely disrupted the prostaglandin biosynthetic and metabolic pathways.
Wait, prostaglandins, like the chemical targeted by Advil and ibuprofen, what is an inflammation chemical doing, building a testis?
I know it sounds weird, but prostaglandins do much more than cause inflammation. They act as vital short-range signaling molecules between neighboring cells. In this context, they're the communication network maintaining that mutual antagonism we talked about. What this discovery shows is that the male testicular environment isn't a switch you flip once in development and then walk away from.
Yeah.
It requires constant, active, ongoing chemical signaling to maintain its male identity and actively suppress the female pathway.
So the cells are literally holding hands, continuously whispering, "Stay male, stay male, stay male," to each other through prostaglandins.
That is a perfect way to visualize it.
And the moment you use enzymes to break their hands apart, the whispering stops, and the default female pathway kicks right back in.
The mutual antagonism is a ceaseless battle. The architecture only exists as long as the cells can physically communicate.
Which is fascinating, but it presents a huge logistical nightmare for our scientists. If they cannot break the factory apart to put the workers inside because the factory will turn into an ovary, how do they get the germ cells into the tubules?
They had to abandon brute force and rely on elegant biology. Instead of breaking the testicular organoid apart, they simply placed the germ cells right next to the intact, fully formed aggregate in a tiny U-bottom plate.
They essentially left the workers on the front porch of the factory,
Basically.
just hoping they wander inside.
Well, not just hoping. They utilized chemical breadcrumbs. The testicular cells naturally secrete a chemokine ligand called CXCL12.
CXCL12, and a chemokine is...
Think of a chemokine as a targeted scent trail.
Yeah.
And the germ cells happen to possess a receptor for that exact scent called CXCR4.
Oh, so the factory is actively broadcasting a help-wanted sign into the surrounding fluid, and the workers have the exact antenna to pick up that frequency.
And it worked flawlessly.
Yeah.
Without any structural disruption to the aggregate, the germ cells followed the chemokine gradient and literally infiltrated the structure on their own. They crawled inside, navigated into the tubules, and settled into their positions.
I love when biology solves its own problems. All right, let's do a third recap before we hit the climax here. Breaking the cells apart feminized them because it stopped the prostaglandin whispering that maintains the fragile male state. To bypass this, scientists use chemokine scent trails to lure the germ cells into the intact factory. So the workers are inside. The researchers let this culture run for 28 days. Walk me through the mechanics of what happens inside that 28-day window.
We witnessed a completely unprecedented maturation process. The researchers tracked the cells using specific protein markers to ensure the germ cells were hitting their developmental milestones. First, the germ cells differentiated into prospermatogonia.
Prospermatogonia.
Yeah, and they verified this by detecting a marker called DNMT3L.
Okay, before we just list acronyms, what does DNMT3L actually do? Like, why is it important that they found it?
Good point. So DNMT3L is an enzyme that helps establish DNA methylation patterns. Basically, it locks in the specific genetic blueprints the cell will need for its future specialized role, and it silences the genes it won't need.
Okay, so it proves the cell is really committing to the process.
Exactly. Following that, they progress into spermatogonia, which was confirmed by the PLZF marker.
And what is the function of PLZF?
PLZF is a transcription factor essential for maintaining the stem cell pool. In a living testis, you don't want to convert all your germ cells into sperm at once, or you just run out.
Right, that makes sense.
PLZF ensures the reserve population is kept safe and self-renewing. Finding it meant the lab-grown organoid was establishing a sustainable long-term sperm production system. Finally, they found SYCP3.
Let me guess, another major milestone.
The biggest one yet. SYCP3 is a protein that forms the scaffolding between chromosomes during meiosis.
Okay, meiosis, the cell division.
Right, the specialized cell division that halves the chromosome number. From 46 to 23 in humans or 40 to 20 in mice, it's required to create actual reproductive gametes. Finding SYCP3 meant the cells were physically preparing to divide and create sperm.
Wow. And what about the factory itself? Did the surrounding somatic cells mature over those 28 days as well?
They did. The somatic cells matured into Leydig cells, which they confirmed by a marker called HSD3b.
HSD3b.
Yes, which is an enzyme crucial for producing testosterone. They also formed paratubular myoid cells, marked by alpha SMA.
And alpha SMA is?
It's a muscle protein. It indicates these cells were wrapping around the tubules, providing the physical structural support and contractile ability a real testis needs to move fluid.
So the tubule is built, the testosterone factory is online, the stem cell reserve is secured, and the cells are having their chromosomes. That is incredible.
But looking good under a microscope isn't enough, right? Did it actually work?
Well, to prove it was a fully functional system, they performed the ultimate test. They took these laboratory-generated spermatogonial stem cells, grown entirely from pluripotent stem cells, without ever touching an embryo, and surgically transplanted them into the tests of living infertile mice.
Walk me through the timeline of that. What actually happened to the mice?
Over several weeks, the transplanted cells engrafted into the host tissue. They successfully reconstituted the entire process of spermatogenesis inside the living mice. They produced mature, swimming sperm.
Wait, really?
Really? The researchers then extracted that lab-grown sperm, used it to fertilize eggs in a laboratory setting, and transferred those embryos to surrogate mothers.
The result was the birth of healthy, living, fertile offspring.
Oh my gosh, I need to take a second to process that. Zero embryonic tissue, just raw stem cells, some chemical steering, a lot of patience, and they generated the entire environment necessary to spark a new living generation.
It's a monumental leap. This raises an important question about the bottleneck of in vitro gametogenesis, which we've struggled with for years.
Because we couldn't make the environment.
Exactly. We struggled to make eggs and sperm in a lab, not because the germ cells were impossible to make, but because we couldn't recreate the intricate, highly communicative environment they're required to mature. This organoid technology bypasses that bottleneck entirely.
Which brings us to the big picture here. Bring it back to you, the listener. Why does a glowing green mouse testis organoid matter to you? Like, what are the tangible applications of this moving forward?
Well, think about conservation biology, for one. We have endangered mammalian species like the northern white rhino that are functionally extinct because there are no viable breeding males left.
Oh, wow.
With this technology adapted, we could take a simple skin biopsy from a preserved rhino, revert it to stem cells, grow an artificial testicular niche, and basically generate an infinite supply of sperm for conservation efforts.
That is a phenomenal application. And what about human medicine?
It opens a completely new frontier for treating human male infertility. Currently, if a man cannot produce sperm due to a failure in his testicular microenvironment, clinical options are incredibly limited.
Right.
This organoid model allows us to study the exact interactions between germ and somatic cells in unprecedented detail. We can literally watch human infertility happen in a dish, test targeted drugs to repair the cellular communication, or potentially one day create patient-specific organoids to restore fertility.
So what does this all mean? It means the black box of mammalian reproduction is finally being opened one stem cell at a time. We are no longer just observing biology, we are learning the language cells use to talk to each other, and we are using that language to build life from the ground up.
It is a profound expansion of our biological capabilities.
It truly is. But before we sign off, I want to leave you with a specific, maybe provocative thought to mull over. based on a tiny nuance buried in this study.
Oh, the meiotic checkpoints.
Exactly. The researchers noted that while the cells successfully started meiosis in the dish, they built that SYCP3 scaffolding, they didn't complete the cell division perfectly prior to being transplanted into the living mice.
Right. The researchers attributed this to what they call stringent meiotic checkpoints. Think of these checkpoints as like microscopic quality control inspectors on an assembly line.
Right. The artificial lab environment was stunningly accurate, but it lacked some microscopic environmental cues required to pass those final strict biological inspections without the help of a living host body.
Which tells us so much about the environment.
It does, and that detail sparks a fascinating question for everyone listening. We just spent this deep dive learning that the male reproductive environment is shockingly delicate. We saw it completely feminized simply because the cells were temporarily separated and stopped whispering to each other with prostaglandins. We saw it struggle to pass strict meiotic quality control outside a living, breathing host. So if the fundamental architecture of male development is this sensitive and this reliant on perfect, uninterrupted chemical communication to maintain itself, what does this imply about how real-world environmental stressors like microplastics, endocrine-disrupting chemicals, modern diets might be subtly disrupting human development right now?
That is exactly the kind of profound question this new technology will finally give us the tools to investigate.
If a highly controlled petri dish is missing a faint whisper, what vital cellular communications are our modern, chaotic environments drowning out every single day? Something to think about the next time you take a painkiller or look at a plastic water bottle.
Definitely.
Thank you for joining us on this deep dive into the blueprint of life. Keep questioning, keep learning, and we will catch you next time.